Today's webinar will be presented by Dr. Ernest Heimseth, who holds a PhD in biochemistry from the Geisel School of Medicine at Dartmouth College. Ernest is a principal scientist in the applications group here at BioTek, a part of Agilent, and today he is presenting on maximizing fluorescent signal using variable bandwidth monocromators for plate reader-based assay optimization. Thank you for joining us today, and with that, I will turn it over to Ernest.
Thank you, Bear, for that introduction, and thank you everyone for tuning in today. As Bear mentioned in this webinar, I will be discussing how to get the most of a fluorescence-based assay using variable bandwidth monocromators. This webinar will be broken down in a few different sections. In the first one, I will be touching upon a few aspects and practices that one should keep in mind when optimizing a plate reader-based assay using fluorescence indicators, such as fluorescent proteins and dyes. I will then be discussing the challenges of working with multiple fluorophores and demonstrate how to optimize the excitation and emission range by building lambda stacks, also known as spectral scanning. I will then demonstrate how variable bandwidth monocromators can maximize your signal, and lastly, how that signal can be refined by linear unmixing. I will then summarize what I discussed and will be happy to answer any questions you may have. So first, let's explore the challenges of working with multiple fluorophores. The important properties of fluorophore, whether a fluorescent protein or dye, are very often distilled down to two wavelength values, which is the excitation maxima and the emission maxima, as well as the compatible filter sets or laser lines at our side. This approach may be fine when working with a single fluorophore. However, when working with multiple fluorophores, it's important to be familiar with its excitation and emission full spectra. And that is the range of light where it can be excited and the range of light where that light can be collected. For example, this is the spectral profile for GFP. The profile with the dotted line shows the range where it can be excited, and the shaded in green profile shows where emitted light occurs. Ideally, you want to maximize the amount of fluorophore you excite as well as the amount of light that you can collect. So when it comes to using optical filters, you would want to place those filters where you can collect the most light. As shown here, as the schematic up top, you have a light source that would emit excitation light. You would have an excitation filter in place. They would refine that light, excite your fluorophore, in this case GFP. That emitted light would then be further filtered with an emission filter, and that would be detected by a detector, either a photomultiplier or a camera. And of course, you would want the excitation and emission filters to be fairly separated so you have no interference between the light collected and emitted. Now, a few words on the emission filters. The filters are given by this value. In this case, it's 485 nanometers with a slash in 20, where the emission is 528/20. All right. Now, what those values mean is that you have a central wavelength of 485, and the 20 is really 10 on both. It can collect 10 plus or minus 10, and the same is true for the emission filter. Is you have a central wavelength that is able to pass through 528 plus or minus 10 nanometers. And this configuration for filters works out well if you're only measuring GFP. However, the challenge comes when it becomes necessary to multiplex two or more fluorophores. Consider YFP, whose excitation and emission spectra is very similar to GFP. So much so that they have overlapping spectra. So you can see here in the dotted orange line where the excitation is ideal for YFP, you also have the emission spectra for GFP as well. So if you consider just the excitation, If you have a sample containing both of these fluorophores, using a filter optimized for GFP excitation, you'll find that YFP is also able to be excited as well, because that light that passes through the filter is able to excite YFP as well. Additionally, the emission filter for GFP will allow any excited YFP signal to bleed through, and it will be difficult to decipher between the two signals. To illustrate this point, consider two cold cultured cell populations, showing these insets right here. One population exclusively expresses GFP and another expresses YFP. Because green and yellow are not easy to decipher, in the left panel I falsely colored YFP to magenta, so you can see that the two different populations. What I hope is obvious is on the right, where you isolated the GFP channel, is those cells that exclusively express YFP are showing up in signal in the GFP channel, and that's because of a mismatch in your filter sets when exciting both of these fluorophores. Now the same is true for YFP's excitation as well, where the YFP excitation filter, which is 500 with a bandwidth of 27 nanometers, as you can see with the excitation for GFP, you're still exciting some of GFP if that's present. Conversely, YFP emission allows GFP signal to bleed through as well, which again it can be illustrated with that same image set. So although less faint, when exciting GFP, you still get some bleed through in the GFP channel, in the YFP channel for GFP cells. Now, this may be easy to decipher and distinguish with images, but when it comes to a plate reader-based assay, you don't have that luxury. All you're going to get is a single value signal coming from that well in a microplate. When in fact that signal is really made up of both GFP and YFP signal. So this illustrated the point that filters will commit you to a wavelength range and bandwidth, as do fixed bandwidth monochromators. As we'll discuss in a little bit, variable bandwidth monochromators enable you to maximize the signal by tailoring the excitation and emission wavelength ranges to the fluorophores being used. And in order to do that very efficiently, the first thing that we need to do is to establish lambda stacks in order to optimize those ranges to get the most fluorescent signal. Now, lambda stacks, also known as spectral scanning, will help achieve the best signal-to-noise ratio. Now, the signal-to-noise ratio is simply your target signal minus your background divided by the standard deviation of your background signal. So the way that lambda stacks work is you would take a fixed, in the case if you were to establish what is the optimal bandwidth and range for your excitation, you would have a fixed emission range. In this case, it's 505 nanometers with a bandwidth of 9 nanometers. And then you would walk across your excitation wavelengths along the spectra of GFP. And what you'll see here is as you get closer to the ideal wavelength for GFP, you'll see an increase in signal. And of course, because you're also encroaching on the excitation of YFP as well, you'll see a slight increase there. Now, again, the signal to noise is simply the calculation of that signal at a given wavelength minus the background at that wavelength divided by the standard deviation. In this case, for establishing the excitation ideal range for GFP, what we see is we get the best signal-to-noise ratio within this ranges of 460 to 475 nanometers. Now, as we establish the ideal range to get the most signal-to-noise ratio for your GFP excitation, you can do the same thing with your emission as well. As you have a fixed excitation wavelength, you can walk across your emission wavelengths and find where is the best signal-to-noise. So as you can see, your signal for GFP peaks around between 500 and 520, and you're going to get background from YFP a little bit further off. So after doing the calculation, what you could find is that your ideal range to collect GFP signal while minimizing YFP background contamination is between 490 and 505. Now, once you've established this for one fluorophore, you could then do this again with another fluorophore. As I'm showing here, you could do this for YFP. You could establish the ideal signal-to-noise ratio range, the range that gives you the best signal-to-noise ratio for the YFP's excitation, as well as build a lambda stack to get the ideal range to get your signal-to-noise ratio for your emission as well. Now that you have this, and you have this range, you can then move on to now optimize the bandwidths of your emission and excitation using your variable bandwidth monocromators, also known as bandwidth tuning. So variable bandwidth monocromators allow you to do two things. They allow you to optimize the excitation range to minimize a secondary signal, in this case YFP, and it also allows you to capture more GFP signal while minimizing GFP contribution. So in this case, what we've done is we've set actually kind of a less than ideal excitation range for GFP at 450 nanometers with a bandwidth of 24 nanometers. And this is because what we want to do is we want to minimize, as shown in this shaded yellow YFP spectra, we want to minimize how much of that is contributing towards collecting light. So what we could then do is increase the bandwidth for emission from 9 nanometers to 16 up to 24. And as you can see on this inset graph, is as we increase that away from the GFP signal, away from the GFP emission spectra, we can increase our GFP signal while minimizing YFP contamination. And what this means is essentially we've able to optimize and increase our signal-to-noise ratio as well. The same thing can be done with YFP as well, is once we've established the ideal excitation range, in this case 518 nanometers, we can then optimize and increase the amount of emission light that we collect for YFP as well. Now the situation becomes even more complicated with working with 3 or more fluorophores, and that's because you have a very limited range if you were focused on YFP signal, from where you can excite YFP and the range where you can collect YFP while minimizing GFP excitation and Alexa Fluor 546 emission as well. However, this can be done, and once you optimize your excitation range for YFP, shown in these shaded columns on the left, you can then expand and build the lambda stack and determine what is your ideal range for YFP emission, even in the presence of fluorophores with very close spectral overlap. In the case here, what we've done is established that you can go as far as use a bandwidth as far as 30 nanometers and still get very robust signal-to-noise ratio. In green, what we're showing is the signal-to-noise ratio of YFP relative to GFP, and in red is the signal-to-noise ratio of YFP relative to Alexa Fluor 546. So now that you've established your ranges for your excitation and emission filters, you can then refine your signal with linear unmixing. And again, this is just to reiterate that filter sets are not ideal when multiplexing fluorophores. However, that's okay, even if filters are your only option, or even fixed bandwidth monochromators. There's still a way to refine your signal. And that's because the signal can be separated by linear unmixing. And what that means is that if you have a signal shown in gray in the left panel here, that signal is really the sum of the signal coming from two different fluorophores. And as the middle and the right panels illustrate, that sum signal, even in different proportions, you can still parse out those two. So as I showed you earlier on, that signal can be separated with linear unmixing, even if you have very close overlapping fluorophores. However, you do need a few things in order to accomplish this. You will need reference samples for each fluorophore. In other words, samples that are pure, either have only GFP signal or YFP, or in these examples, Alexa Fluor 546. And ideally, you would want to perform linear unmixing after you optimize the excitation and emission ranges using lambda stacking. And as an example here, what I've done is demonstrated this using cell lysates from cells expressing either GFP, YFP, or a purified dye, Alexa Fluor 546. And as you can see in the first three columns of this graph, these are the signals coming from these standards. However, if you were to mix them in some proportion, as in the gray bar, what you can see is it's difficult to decipher. However, once you establish a standard curve based on your references, you can parse out what you can unmix the GFP signal even when multiple fluorophores are present. Similarly, you can do this with YFP as well, where as long as you have references for a reference sample for YFP, GFP, and Alexa Fluor 546, you can have a mixed sample and still be able to distinguish out the signal of interest, in this case YFP. Now, to wrap up, what I showed you is some of the difficulties working with multiple fluorophores and a way to bypass those kind of challenges, which are to establish the ideal excitation and emission ranges using lambda stacks, as well as linear unmixing. And just to point out that variable bandwidth monochromators give you the flexibility to do two things. It allow you to optimize the range of your target fluorescent signal, and also to capture more of that target fluorescent signal as well. And it's important that when deciphering the signal of multiple fluorophores, the excitation and emission range is more important than the bandwidth. Narrower is not always better, as we showed with the GFP. Is it's positioned correctly, a greater emission bandwidth will actually help you get a better signal to noise. Filters, as well as fixed bandwidth monochromators, do commit you to a wavelength range and bandwidth. However, using linear mixing still provides a way to get the most out of those signals as well. However, ideally, variable bandwidth monochromators enable you to maximize signal by tailoring the excitation and emission wavelength ranges to the fluorophores being used. And we have multiple plate readers that are capable, that have these capabilities. And newest to the product line is our Synergy H1 launched this summer, that in addition to filter capabilities now has variable bandwidth monocromators, as well as our Synergy Neo 2 and Citation lines as well. And with that, thank you very much for tuning in.
Great. Well, thank you, Ernest, for a wonderful presentation, and have a great rest of your day.